Okay, so say hello to the people online who are still not here. Why aren't they here? We don't know. Okay, so let me shrink it down. So let's take a minute and set up your page if you haven't already. So set up your page for the notes. Again, you guys online, if you need to pause this whenever you need to. If you have questions, you need to send them in on Remind. Don't send them in on email through Google Classroom because I don't even read those. Did you guys ever read your Google email? Yeah, most of us don't. It's the only, we just have it for the Google Classroom there. Yeah, you guys know, don't ever send me anything on an email like that. I read my school email, so if you have my school email address, that's fine, but not through Google Classroom. I have enough email crap. I don't want any more. How do you get in touch with me? Through what? The Remind app works fine. And it limits you to a number of characters, so you don't get too wordy on me. Sometimes, you guys. Okay, so this is Unit 11, DNA and Protein Synthesis. What is today? 8. And this is period 7. Period 7 is the best class period, right? Yes. Okay, so we're doing B6C, and this is part 2 here. So explain the purpose and process why, and process of transcription and translation. Using, and here we started making these yesterday. Did we start coloring yesterday in here? Okay. Using models of DNA and RNA. And at the end of the class period, I'm going to show you a video. I'll put it on Google Classroom too, but I'll show you a video. And the main reason I want you to see the video is it's one thing to see it all and talk about this all in words, and it gets pretty complex. It's another to see it. It makes it clearer and actually simpler when you see an animation of what it is we're talking about here. Okay, so yesterday we focused on transcription. Today we're going to focus on translation. So the title for this is Translation. This is what we're focused on. And I'm going to put protein synthesis in parentheses because remember, both of these two pieces, transcription and translation, together make protein synthesis. Protein synthesis, again, is just making what? That's it, making proteins. That's all we're talking about here. So we're going to look at translation then. And let me make this just a little bit bigger so it's easier for you guys to see. Okay, so translation, that word means to change to a different language. You guys should know that already, right? Sometimes when they're talking about protein synthesis, they also say it means like to decode something. So it means to change to a different language. If I could spell language. Or decode. We talk about the coded message in the DNA. It's kind of like, spy messages where, they have all sorts of weird letters, and then you can, if you know what the code is, you can figure out what it says. That's kind of like what we think of with the DNA. All those nitrogenous bases, A's, G's, C's, and T's, they're like a secret message, and we can uncode it or decode it with the RNA, and we can change it to the language of the protein instead of the language of the nucleotides or the nucleic acids there. So it means that we're going to change the language, and we have a purpose. And when I say purpose, what I'm talking about here is the end product. Okay, so I'm going to backtrack the page here. And if you remember, I try to kind of keep some things consistent. We said the purpose, or the end product, right down here, is to create RNA when we're talking about transcription. So now when we talk about it, we've got the purpose or the end product in translation. And our end product, what we actually make, is the protein. So when we finish with translation, we'll have a protein. When we finish with transcription, we had a messenger RNA, mRNA. When we finish with translation, we'll have the whole protein there. So it has to happen, though, together. So here we have the process. Okay, and on the last one, I just jumped in and gave you some steps here, but I want to give you a little bit more information because it's a little bit more technical in its explanation. So the first thing I want you to make sure you realize is that this occurs in the cytoplasm. Where's the DNA? In a cell, where's the DNA? In the nucleus, right? I don't think I wrote down yesterday that it occurs in the nucleus because I just made the assumption that you figured that out. But if you want to do this, you could write down in nucleus. I just assumed you guys figured that out, but I didn't kind of write it down here. I think it's obvious if I asked you, would you be able to tell me translation is in the transcription's in the nucleus and translation's in the cytoplasm? Yeah? Okay, so transcription, when you write it down, you're copying down what the DNA says, so it's got to be in the nucleus with the DNA. Translation occurs in the cytoplasm. Where in the cytoplasm does it happen? Ribosomes. Do you remember what ribosomes are good for? That's where proteins are made, right? The ER, the rough ER, isn't that where proteins are made? Yeah, that's what we're making right here. So it's in the cytoplasm, specifically it's on the ribosomes, right? And the ribosomes, not all ribosomes, but a lot of them are hooked to the ER, right? Some of them are floating around there, on their own, but a lot of them are hooked onto the ER. Okay, so we have some terms that you have to know. So the next piece here I'm going to put is terms. Because biology is vocabulary intense. And it has a lot of words that are either uniquely defined, so that when you go into an English class, it's like, oh, that's not what we learned in biology. Or totally new words that you've never in a million years put together, like mRNA. 4 letters. It's not a word, but it's something that we use as a word pretty easily. So the first one I'm going to talk about is lowercase m, capital R, capital N, capital A. So mRNA. That's how it's written. There are lots of types of RNA. We're going to hit 3 today, and those are the only ones you're going to have to know about. mRNA, rRNA, tRNA. So mRNA stands for messenger. Messenger, there's your M. So right here's your M. Messenger RNA carries the DNA instructions, or the message. You cannot take the DNA out of the nucleus. It needs to stay safe and protected inside the nucleus. Because if it doesn't, then you have the possibility of messing up the DNA. And everything, if you have messed up DNA, everything made after that is going to be messed up, right? We don't want to mess up the DNA. We want to keep it. Every time that cell, if you mess up the DNA, every time that cell divides and makes new cells, they're going to have messed up DNA, and you don't want that. You keep that DNA inside the nucleus as safe as possible. So we're going to go into the nucleus, we're going to make a copy of that DNA, that's what transcription is, and then that messenger RNA carries that DNA out of the nucleus into the cytoplasm. When I say instructions, if you think of it as a message, like, hey, this is how you do it, here's your message, here's what you need, this is your instructions. Yeah. The next one is rRNA. rRNA is ribosomal. And RNA is really kind of an interesting substance here. There's your rRNA. Is that for me? Or is it the other room over there? Yeah. Okay, so ribosomal RNA makes up the ribosomes. What are ribosomes made of? rRNA. They are not true membrane-bound organelles. They don't have any membranes around them. They're just a chemical RNA wadded all up into a little shape. They're really weird. And RNA's crazy stuff. RNA can actually work as an enzyme too, which usually we think, oh, proteins, they're enzymes. Yeah, a lot of proteins are enzymes, but this nucleic acid, this rRNA is also an enzyme. You get some weird stuff going on here, so it's pretty complex stuff here. So rRNA is the ribosomal RNA. It just makes up the ribosomes. Okay. The next one, and I'm going to kind of go off track of the RNA, let's look at the ribosome. The ribosome is just the site of the protein synthesis. If I use the word site, do you guys know what that means? Site? What's it mean? Yeah. It's the site of protein synthesis. Again, most of the time it's going to be hooked to the ER, but not always. You've got just these little ribosomes floating all over the place too. Now let's get back to the other type of RNA, and that's tRNA. And tRNA stands for transfer. So this is the one, what are proteins made of? Amino acids. We haven't said anything about amino acids yet. Something has to bring the amino acids in, and that's the job of the transfer RNA. So the transfer RNA carries the amino acids. Can I put AAs? Yeah. Okay. So the transfer RNA is bringing in the amino acids, because we have to make that protein, we have to put the amino acids in order, and we got to hook them together. Because proteins are made of amino acids, so if I ask you, what do you call a chain of amino acids, what are you going to tell me? A protein. Yeah. That's what they are. You hook a bunch of amino acids together, you get a protein. So a chain of amino acids is a protein. The order of the amino acids tells you what kind of protein, what protein it is. If you change the order of the amino acids, it's a different protein. So maybe brown eye color instead of blue eye color, or green eye color, or what have you. The last word, and again I'm going to pop this in, you guys should know this, is protein. Protein, okay, so here's another word. I think you've heard it a couple of times or maybe seen it, but a protein is also known as, aka, a polypeptide. So the bond between two amino acids is called a peptide bond, and so that's why they say a polypeptide. So if they say a polypeptide, they're just talking. It's another spiffy word for a protein. So don't be freaked out when you see that. If they say polypeptide, somewhere in there they're going to mention protein. So you don't have to memorize that, but just don't be freaked out when you see those words. A lot of times when they throw words at you on the STAR test, you're thinking, crap, I've never heard of that before. Well, don't choose that as an answer, okay? Because they're going to throw some at you on purpose like that. Okay, so remember that proteins make up most of your body parts? You could think of it another way. Instead of saying body parts, they make up most of your traits or characteristics, right? If you want to add in there body parts, that's okay too. So you're either mostly made of protein, or the protein is directing how your other parts are made. Like your bones are not mostly protein. They're mostly this chemical called calcium phosphate. They've got some protein in it too, but it's mostly calcium phosphate. It's the proteins that are controlling how the calcium phosphates are put together. So it's all about the protein, okay? And then they're made of amino acids. You already know that one. And the order of the amino acids determines what protein it is. So the order of amino acids determines the identity of the protein. If you mess up the order of the amino acids, yeah, and I don't mean on purpose, like, oops, it's a mess-up mistake. Typically, that's going to mess up the protein. Maybe it'll just be such a minor mess-up that it still works okay. Maybe it's a mess-up that's really going to make it so the protein won't work like it's supposed to anymore. It just depends on what it is. Okay, so let me grab another color here. Let's do brown. Okay, so on the other one, on transcription, I gave you the purpose and the process and did numbers. So now let's look at the numbers here. So let's start with the process and let's do number 1 here. It doesn't look like a different color, but it is. That was a purplish color. This is a brownish color. Okay, so let's start with the mRNA. What's going on with the mRNA? Well, the mRNA, you just made the mRNA in the nucleus, and it's coming out to the cytoplasm. So the mRNA with the info from the DNA is this big enough, or do I need to make it bigger? That's good. Okay. So the mRNA with the information from the DNA moves out of the nucleus, and you know there's stuff that happens to it inside before it's ready to go. It has some really crazy stuff happening to it. Like when you copy off a section of the DNA before the mRNA is ready, it gets edited, which is crazy. They just found this out fairly recently. So pieces of it are cut out, which then you think, well, how does it know what pieces to cut out? And it does. I don't know how it knows. They're still working on all that stuff, but it's kind of cool that craziness happens to it like that. It moves out of the nucleus and attaches to the ribosomes. What's a ribosome made of? There you go. Ribosome, and I might put that up here, made of rRNA. See the rRNA makes up the ribosome. So remember that ribosome is just rRNA. So we use all three types of RNA for this process. The second thing to happen is that the mRNA, okay, so it's not necessarily happening, but I got to take you through the steps. In the mRNA, every 3 nitrogenous bases is called a codon. They're triplets. They come in 3s. So in mRNA, each set of 3 letters, does that make sense? Each set of 3 letters, you know what I'm talking about when I say that? Each C, G, A, whatever it is, each set of 3 letters is called a codon. So it's like all the words that are in there are 3 letters long. So it's a secret code. All the words are 3 letters long. And there's a complement to that. Remember one side of the DNA, and you can build the complementary side to that? So here we've got the RNA, we can build a complementary side to that. And what we do is we use the tRNA. In tRNA, in tRNA, each set of 3 letters is called an anticodon. So we have the codon and we have the anticodon. It's opposite. It's complement. So these two are going to hook up together ultimately. That's how they match up. And this anticodon right here determines which amino acid that tRNA carries. So the tRNA, each 3 letters in the tRNA is called an anticodon, and that anticodon determines the amino acid that the tRNA carries. It is the tRNA. So there are going to be 3 letters that are exposed on this transfer RNA. Think of the transfer RNA kind of like a truck. It's going to carry around the amino acids. One transfer RNA can carry 1 amino acid, and that's it. And so it's got 3 of those letters exposed, and those 3 chemicals, those 3 letters that are exposed, allow it to hook on to a particular amino acid chemically. So it's all chemically controlled. It's craziness. Okay? And those 3 letters are going to match up with one of these codons here. Okay, number 4. The tRNA anticodon matches to the mRNA codon. In the presence of the ribosome, this is the only place it's going to happen. Now, the ribosome acts like an enzyme. This could happen outside of the ribosome, but it happens so slowly that it's like, let's just ignore that crap. So the tRNA anticodon matches to the mRNA codon in the presence of the ribosome. That's really important. So this ribosome acts as an enzyme. Yeah. So that ribosome acts as an enzyme. Let me move this down a little bit more here. Okay, is that good? You need to move it down more? Okay. Okay, so that tRNA is going to hook up with the mRNA in the presence of the ribosome, because that ribosome is going to act as an enzyme. It's going to make that chemical reaction happen that goes on in there. That tRNA right here brings with it its amino acid. Brings its amino acid. And this is how the amino acids are put in order. So you've got this big long line of an mRNA. Part of it's in the mouth of the ribosome, kind of holding it in place. And the stuff that's in the ribosome, the first tRNA brings in its amino acid, sits there. The next three spots, a different tRNA comes along and brings its amino acid in there. When those two amino acids get together, close enough together, they form a bond. Now, it's got to be in the presence of the whole ribosome that it does that. But when they get close enough together in the presence of the ribosome, they form a bond. Now you've got two amino acids hooked together. And then a third transfer RNA comes in with its own amino acid, gets together in the ribosome. Those two hook up with that one. Now you've got a chain of 3 amino acids. The first one leaves, munches down a slot, and another one comes in. So it just keeps building and bringing in, the transfer RNAs keep bringing in the amino acids here. So 5, the amino acids bond together in the presence of the ribosome. They don't bond much outside the presence of the ribosome because it just requires so much energy for that to happen. And they form a chain. So the amino acids bond together, forming a chain. What do you call a chain of amino acids? A protein. Okay, there's one more piece of information we need to put on here before I show you some videos. And again, I'll post these online. I strongly suggest you watch them. The animation, even if you turn the noise down, watching the process of it, that animation is really important because you're going to have to explain it. That's exactly what the tick says. Explain the purpose and the process of transcription and translation. So on the test, you're going to have to explain this. Okay? So the last thing is, when we talk about those codons and anticodons and the amino acids, there's a chart that we have to use. Because there are only 20 amino acids that most mammals use. So use a chart to determine which amino acid matches each codon. Or codon. It just depends on what you're talking about, whether you're talking about in terms of the mRNA or in terms of the tRNA. So we can put codon or anticodon. Either one will work there. A lot of times what we do is we work with the codon, and we kind of work backwards from that. So I want you to see this chart. So when you have that, this is the chart I'm talking about. And I'll put a couple of examples of the charts in here for you guys. We're going to use it next week. So I'm going to pop this back up here a little bit. So here is a codon chart. And so let's say that my first codon said UAG. So first codon, UAG. So I'm going to start over here on the left. So here's U. And then it says the second letter is A, so I move over to A. And then my last letter is G. So UAG, that's a stop sequence. That means it tells it, hey, we're done with this protein, stop. Let's say it's AUG. So I go down here to the A, and my second letter is U, and then my third letter is G. Find it in here, and oh look, there's the start sequence. So when it comes across these 3 letters, they're saying, hey, start making your protein right here. Let's say it's AAC. Okay, AAC. So we'd go to A, and then go across to the second A. Second A is up there, and then the third letter is C, AAC. That's the amino acid that it puts there. Asparagine. Yeah? What if it's UGC? So we have U, we go over to G, and we go up to C, and I don't know why they didn't put anything there. That's something's wrong there. Okay, so let's say it's AGU. We put the 3 letters together, it's AGU. There's the amino acid that would go in that place. So that's what you would do. Whatever those 3 letters are, it's like a secret message. These 3 letters mean this. Put that amino acid there. Okay? So here's what I want you to do. On page, is it 3 or 5? Page 5. So page 5, I have 2 pictures left on this page. And what I want you to look at